So we know that blood is divided into plasma and cells. About 45% of the blood is going to be the cellular component, the red cells and the white cells. The vast majority of that, of course, will be the red cells. And we know that blood is also divided into the plasma. But on this talk, we want to think mostly about the cells in the blood. So we have blood and we have cells in the blood. So what are the cells in the blood? Now there are two main types of cell in the blood. There are the leucocytes, which are the white cells, and they are the erythrocytes, which are the red cells. So leuco means white, and in the UK we spell this with a C. In the States it's often spelt with a K. So leuc means white, leucocytes literally white cells. Erythrocytes, erythro means red, so literally the red blood cells are the erythrocytes. Now there's going to be about 5 million red blood cells per cubic millimetre of blood. The vast majority of the cells in the blood are the erythrocytes, the red cells. And these cells live for about 120 days after they've been released from the bone marrow, from the red bone marrow where they're produced. And they circulate in the blood for around about 120 days. So here we see a red cell. And what we notice is the red cell is a biconcaved disc. It's darker on the outside and it's lighter on the inside because they are biconcave discs. And if we were to look at those in cross-section, they look a bit like this. They're thinner in the middle. And they're about 7 micrometers in diameter. The red cells, the erythrocytes. And of course, as the red cells pass through the lungs, they pick up oxygen. The oxygen combines with the hemoglobin, which is the red pigment in the red cells, forming oxyhemoglobin, increasing the saturations of the blood. The cells then circulate to the tissues. The oxygen is liberated from the oxyhemoglobin, restoring the hemoglobin to its reduced hemoglobin state. Sometimes that's called deoxyhemoglobin, and that will circulate back to the lungs to be reoxygenated. And the red cells are very flexible cells. They can squeeze down to get through very small capillaries. So for example, in the brain and in the kidneys, there are very small capillaries and they can actually squeeze down to get through. After 120 days or so lifespan, they're broken down by macrophages, largely in the liver and spleen. They're broken down and the hemoglobin is recycled in the form of bilirubin to form bile. So it's not wasted, it's used as bile. The protein components are reabsorbed. So the red cells are being produced all the time in the red bone marrow. And of course, in the circulation, the red cells do not have a nucleus. They are the only cells in the body that are living cells, they are metabolizing cells, but they don't have a nucleus. Now, when they were in the bone marrow, when they were forming, they did have a nucleus, but the mature cell doesn't. And actually, for the first couple of days, when the red cells have just been released from the bone marrow, it's still possible, if they're correctly stained, to see some nuclear fragments in those very, very young red cells when they're only a day or two old. And these very young red cells are called reticulocytes. So that's the erythrocytes. Next we think about the leucocytes, the white blood cells. Now people started looking at these a long time ago under rather primitive light microscopes, and that's when their names were generated. So blood samples were taken, they were stained, and the cells were examined underneath the light microscope. So the way we classify these is based on this early observational data. So the leukocytes, there are a class of leukocytes, that is a class of white blood cell called the granulocytes. And the granulocytes are so called because they have visible staining granules in their cytoplasm. So granulocytes, the cells with granules. And the most common one of these are the neutrophils. Then there's another type called Eosinophils. And there's a less common type called Basophils. So you might have noticed already that all of the granulocytic leukocytes, so these are leukocytes, but they are granulocytes, so they are granulocytic leukocytes, they all end in phil. Neutrophils, eosinophils, basophils. Now, as we've said, the most common form are the neutrophils, and the neutrophils have a lobed nucleus held together by thinner strands. And on a blood slide you might observe 3 or 4 lobes in the nucleus. And for this reason these are sometimes called polymorphonuclear cells or polymorphs, because poly means many, morph is the shape. They're polymorphonuclear cells. And these have granules that are visible with a light microscope after the cell has been appropriately stained. They are granulocytic leukocytes, and they are polymorphic. They are polynuclear cells, polymorphonucleocytes. But we normally call them neutrophils. Particularly important for fighting bacterial infections because they can phagocytose bacteria, and indeed they can phagocytose dead tissue cells as well. So the neutrophils are absolutely vital for protection against bacterial infection. Now the eosinophils have a 2-lobed nucleus, something like this, 2 lobes held together by a thicker strand. So these are the eosinophils, and these have tend to have larger granules. And the eosinophils are particularly important for fighting parasitic infections. So infections with worms, for example. The eosinophils will combat that. So we've got neutrophils, eosinophils, and the third class of granulocytic leukocyte we have are the basophils. And the basophils again have a 2-lobed nucleus, that kind of shape. Although very often it's very hard to see the nucleus in the basophil because of the large granules filling the cytoplasm. Now the amount of basophils in the blood is very low, but what the basophils will do is they will migrate into the tissues, and in the tissues we call basophils mast cells, and these help to facilitate the inflammatory response. So in the respiratory mucosa and in the skin there's a lot of these basophils. And with an appropriate stimulus they'll release histamine, which will cause an inflammatory response. This is why we give antihistamines for some inflammatory, particularly allergic disorders. So, cells, erythrocytes, leukocytes, granular sites, neutrophils, eosinophils, basophils. The next classification of leukocyte we want to think about are the agranulocytes. The agranulocytes. Now, A means without granules. Now, it's not that these cells don't have granules, it's just that people couldn't see them under the light microscope, because indeed some of these cells do have granules, in fact. So when we're thinking about the agranulocytes, there are monocytes. Monocytes are fairly large leukocytes. They are agranulocytic leukocytes present in the blood, and they have a fairly large kind of kidney-shaped nucleus like this. And the monocytes in the blood are very important for combating chronic bacterial infections, such as tuberculosis, for example. But the key thing about the monocytes is they don't stay in the blood. The monocytes can migrate out of the blood vessels into the tissues. And in the tissues, the monocytes will become macrophages. Now, macro means big. Phage means to eat. These are the big eaters. They will phagocytose bacteria. They will phagocytose dead tissue cells. And they will also release numerous cytokines, chemical messengers released by the macrophages that coordinate processes such as inflammation and wound healing. So absolutely vital that there are these macrophages in the tissues. And we also now know that monocytes can differentiate into dendritic cells. Now, the dendritic cells are like, they have, kind of like octopuses really, they have big long things coming off them like that. Tentacles coming off them. And that means if any antigens come along to there, or come along to the other side of it, then these can detect them. They are antigen detecting cells. So they might detect viruses, they might detect bacteria, and if they are detected, they will migrate to the lymph nodes and present this antigen to the other immunological cells present in the lymph nodes. So they are antigen detecting and they are antigen presenting cells. And they both differentiate from the monocytes. So the monocytes can become macrophages or dendritic cells once they are in the tissues. Now, another cell fragment that we get in the blood are the thrombocytes. Now, the thrombocytes are cell fragments. In the bone marrow, there's a large cell called a megakaryocyte. And the megakaryocytes break up into hundreds of little fragments, and these fragments are the thrombocytes. And thrombocytes are platelets. It's two words for the same thing. Thrombocytes and platelets means exactly the same thing. And these are absolutely essential for the process of blood clotting. So if a blood vessel is cut, first of all the platelets or the thrombocytes will come together to form a platelet plug, to form an initial hemostasis, and then they can initiate the full coagulation cascade to cause full blood clotting. So the thrombocytes absolutely essential in that process. Now, we're still on agranulocytes. I have to make a bit of space here. Rub off our granulocytes. So the agranulocytes, monocytes, thrombocytes. The other main classification of the agranulocytes are the lymphocytes, the lymphocytic leucocytes. Still white cells, still agranulocytic white cells, but these are the lymphocytes. As the name suggests, these can be associated with lymphatic tissues, but they're also present in the blood. And we can identify two forms of lymphocyte. There's the large and the small. That's quite easy. Now, the large ones are the NKs. The large lymphocytes are NKs. Do you know what NKs stand for? It's a great name. The NKs are the natural killer cells. The natural killers. So these large lymphocytes will kill cells of the body. Why would you want to kill cells of the body? Well, if those cells of the body are infected with viral particles. So viruses are obligate intracellular parasites. The viruses don't breed, they don't reproduce independently like bacteria do in the tissue spaces. They have to be inside a cell. They have to hijack the genetic apparatus of the cell in order to reproduce. So the NK cell will come along and it will just completely kill the virally infected cell. And of course killing all the viruses that are in it at the same time. And also NK lymphocytes can kill cancer cells sometimes. So as long as the natural killer cell can recognise that the cancer is abnormal, it can eradicate malignant tissue. So the large lymphocytes are the NKs. Now the small lymphocytes all look much the same. They've got a very large nucleus, relatively small area for the cytoplasm around the outside. And the small lymphocytes can be divided into B and T lymphocytes. Now the B lymphocytes are so called because they mature in the bone. The T lymphocytes are so called because they originally mature in the thymus gland, in the thoracic cavity. Now there are different types of T cell. So there are T helper cells. There are T suppressor cells. And there are T cytotoxic cells. Now the T helper cells, what these do is they help the B cells. So the T helper cells help the B cells to produce antibodies. That is the immunoglobulins, the specific immune proteins. The T suppressor cells inhibit that process and decide when the infection is finished, when it's time to stop producing large amounts of antibody. And the T cytotoxic cells, a bit like their big brothers and sisters, the natural killer cells, these can kill virally infected cells, and also they can kill cancer cells, which of course is great when that happens. So the lymphocytes can be large or small. The small can be B or T. The T can be helper, suppressor, or cytotoxic. Now it's the B cells that are actually helped by the T helper cells to produce the antibodies, the immunoglobulins. So the B cells are producing the immunoglobulins. The specific immune proteins that give us specific acquired immunity. Absolutely vital. So for example, if someone has HIV infection, human immunodeficiency virus infection, the HIV virus primarily attacks the T helper cells, reduces the population of T helper cells, meaning the T helper cells no longer help the B cells to produce the immunoglobulins, meaning the patients become immunocompromised, get infections, and of course tragically that can include life-threatening infections. But when there is an infection, the B cells proliferate, there's a process of proliferation, and you get millions or billions of these B cells produced in order to produce huge amounts of immunoglobulins, and these are called the plasma cells. So if someone had a viral infection, for example, you would expect the number of plasma cells to increase so that huge amounts of immunoglobulins can be produced to get rid of the infection. And then when the infection's gone, another type of B cell is called a memory cell, a memory B cell. And that remembers how to make that specific immunoglobulin against a specific antigen, meaning that the immune response will be much quicker the next time. So the agranulocytes are the monocytes, thrombocytes and lymphocytes. The monocytes in the tissues can divide into macrophages and dendritic cells. The lymphocytes can be large, which are the natural killer cells, or they can be small. The small can be subdivided into the B or the T. The T can be subdivided into the helpers, suppressors and cytotoxics. And the B lymphocytes can be subdivided into plasma cells and memory cells. So I really hope your piece of paper is a lot more tidy than my board, but that gives us an insight into the initial classification of the white blood cells and the red cells.